Paul Brown. Tonight we were tasked with the opportunity to record another episode, and this time we're going to talk about DNA. So how could we explain this as it's 6 sections to bring young ones from 2 to 7 or 8 up to speed with what the chapter entails? The assignment is kind of lengthy this time. It's more detailed. So I want to break down these sections, but when asked what DNA was, we had to relate that to something they would kind of grasp to. And so the thing that we used as our bait tonight was 2 different concepts. The first concept was, I thought about the 2 channels that they watch more than anything. 2 shows. One is Daniel Tiger. Daniel Tiger is a depiction of a lion like Mr. Rogers when we were younger. Another thing is Disney Plus. They love Disney Plus. And so I thought about that instead of, we would have the Disney Channel, and DNA would be Disney's next attraction. Or it could be Daniel's new adventure. And so when we put it in terms that they could relate to, then it became interesting. And so I told them, instead of Walt Disney, we'll start off the historical basis of modern understanding about DNA with Frederick Miescher. And Miescher came up with this concept, 2 discoveries: nucleic acids and the development of the double helix model that we know. And of course, science builds upon science. So about half a century later, we saw that DNA, it was kind of passed down to a young bacteriologist named Frederick Griffin. He showed the scientific community that there was a thing called bacterial transformation. So later on, a group of scientists, Oswald Avery, Colin McLeod, and Macklin McCarty, they took the transformation process further by showing that DNA actually has a transforming principle. So when they would do experiments, they started to notice that it wasn't other things that they had suspected that was causing this amazing transformation. It was actually the DNA that they were seeing make a transformative change within things. Please forgive my yawning. This has been a very, very long day. The second chapter was about DNA structure and sequencing. So I tried to just grab things and make it into something that they could actually understand. So the first things I thought about was the nucleotides. And we use that as Lego blocks, things that they always enjoy to play with when they're at our home. The 3 layers are blue block, nitrogen. Those things are very interesting. They start to ask what was nitrogen. Then we have the 5 carbon sugars. Of course, I took just 5 different types of sugars that we have in the house. My wife doesn't use sugar, so I had to get some of my treats and show them that this is the concept of the 5 carbon sugars. There was a phosphate group. There are purines that are double ring structures. There are also, I hope I pronounced this right, pyrimidines are smaller. And they're also a 6-member structure within the DNA. My 3rd chapter talked about the basis of DNA replication. There were 3 types: conservative, semi-conservative, and also dispersive. The conservative was relating to something parental, where the parental DNA stays together. The daughter strands, they stay together. The semi-conservative, we spoke about how each of the 2 parents' DNA, then you have 1 old strand. So they started to see the parental concept of DNA. And of course, we're thinking about Daniel's next adventure. The dispersive told us that both copies of DNA have double-stranded segments of the parental DNA, and newly synthesized DNA that's spread around. So that was very interesting to see, very interesting to learn. I always feed off their energy. So we kept getting into the DNA replication when it came to prokaryotes. And so there was this like 8-step process that we saw. DNA pop 1, DNA pol 3, helicase. You had ligase. You had, I guess I'm gonna pronounce this wrong, primase. Hopefully that's right, Professor. You had sliding clamp. You had topoisomerase. Topoisomerase. Forgive my Virginia. It's hard to pronounce certain things. I'm sorry. And also the single strand binding proteins. They were very interesting, which binds the single stranded DNA to prevent DNA from rewinding back. Each process was very just fed off the next, and we kind of built that into a neighborhood adventure where the DNA pol 1, how it removes RNA, replaces it with new synthesized DNA. The DNA pol 3, how it's the main enzyme that adds nucleotides. The helicase opens the DNA helix by breaking hydrogen bonds between the nitro. The 4th step was seals the gap between Okazaki fragments to create one continuous thing. We also had the primase, which synthesizes RNA. Primers needed to start replication. The sliding clamp helped to hold the DNA. Polymerase in place when nucleotides are being formed. We also had the topoisomerase, which helps relieve the strain on DNA when unwinding by causing breaks, revealing the DNA. And of course, we already spoke about the single strand binding proteins that binds to that single stranded DNA to prevent DNA from rewinding back. On my 5th segment of the chapter, which was the 5th adventure, we went into DNA replication in eukaryotes. This replication is essentially very similar, almost the same as prokaryotes, but there are some differences within the origin of the replication. One has single, the pro has single, the U has multiple. The rate of replication, which is important. The pro, of course, is 1,000 nucleotides, and the U is 50 to 100 nucleotides. DNA polymerase types, one had 5, the U had 14. Telomerase, one wasn't present in the pro, one was present in the U. The RNA primer removal that came from DNA pol 1 for your pro. For eukaryotes, it was RNA, SEH, strand elongation, DNA pol 3, pol A, pol 2, pol 3. So it was a very interesting way to try to learn this, but also to explain this. So I was very excited about that. The final segment talked about DNA repair. There were 3 things that were very important within that. That was proofreading. Proofreading in DNA repair is when the DNA pol reads the newly added base before adding the next one. The second part of DNA repair was mismatch repair, when errors are corrected after replication is completed. Our 3rd step, was DNA repair, was nucleotide excision, which is similar to mismatch, except it's used to remove damaged bases instead of what mismatch does. It pretty much corrects it after replication is completed. So in summarizing our chapter, it was very, very interesting to see how all things kind of taught us something in a progressive nature. We talked about the history of modern understanding when it comes to how DNA had its beginnings. We learned about DNA structure and sequencing, the basis of DNA replication. Very important. DNA replication in prokaryotes. We also had DNA replication in eukaryotes. And then there was DNA repair. And so, as we learned all of that as a collective, one had to think about what kind of experiment or what type of experiment could we do where it would be something where it wouldn't scare the bejesus out of children, but it would also be something that they could kind of grasp and understand without the brutality of seeing blood and seeing an experimental animal die. And so I had a friend of mine on my bookshelf, on bookcase, that I have a library in our home. So this is our experimental guinea duck tonight. This is our duck. We're not going to advertise who it's for, from, but this is our guinea duck. Our guinea duck is going to be our experiment that was conducted by none other than our bacteriologist friend and scientist, Frederick Griffin. But tonight, Paul Brown is the scientist. I have on my Paul Quinn. Also, it's football season, so let's go Tampa Bay. It's very important as a scientist to know what it is that I'm trying to do to show you how an experiment is successful in building on to the next thing that's coming within the scientific community, especially within biology that we love. We love biology. It's our favorite class. And so one thing he did that was interesting to me, and the children also found it very interesting, was the living S cell, the living R cells, and the heat-killed S strand. And so this is our syringe. As you can see, the syringe is full of living S cells, pathogenic controlled, inside of our syringe. We're going to inject Ducky, like so. Ducky is quack, 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 boom, dead. Our next candidate, we have another duck, Ducky number 2. Ducky number 2 is injected with living R cells. They're non-pathogenic. Booka, quack, 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 quack. As you can see, Ducky number 2 is still alive. Now, our third candidate, boom, let's use the heat-killed S strain, and a live R strain. What happens when we do this? Let's check. Injection. Quack, quack, quack. The duck dies. And so one of the things that we realized while conducting this experiment is the ducks died during 2 of the 3 instances. They died because they were injected with something that would kill them. When they were injected with something that wouldn't, they lived. And so in closing, thank you so much, Professor, for always bearing with my videos. I love to be creative because this has become my favorite subject of learning. Thank you so much for the opportunity every week to have another episode of Paul Brown Biology with Paul Brown. Good night, and you all be safe, and go Quinnite Nation.